All right, and I will go ahead and share my screen. Yeah, that screen. All right, can we see it?
Yes.
Yeah, awesome. All right, well, welcome everybody. So I am Megan Heafy. I will be guiding you today a little bit. So welcome to our session called Hands-on Biotech Education in the Current Hands-Off World. I am at the BioEd Institute, and we have somebody from Mini PCR joining us. So yeah, there's Allie. So we'll do introductions as we go. If I can advance the slides, there we go. So again, my name is Megan Heafy. I'm the Equipment Depot Manager and the Project Coordinator at the Georgia BioEd Institute. So as you can see here, my email is in the bottom. I'll put it in the chat a little bit later too. So if you guys have any questions, feel free to reach out and I'll connect you with Allie or Janet, who you'll hear from in a little bit as well. So I'll let Kristen introduce herself and then we'll move on.
Hi, I'm Kristen Boskin. I am the director at the Georgia BioEd Institute and get to work with Megan on a daily basis. Thanks you guys for joining. This is a long day. I know you've had class all day, but really happy to see you all here. And I see some familiar faces. I see Elizabeth and Emily Strickland and a few other people that from the previous training last year. So good evening.
Yeah, welcome everybody.
So I'll just start off a little bit about our organization. So we are the Georgia BioEd Institute, and we are a part of a greater organization that you can see there at the top, which is the Center for Global Health Innovation. And there are four other branches within there, or sister divisions, if you will. So Georgia Bio is basically, they are working toward public policy advocacy for anything that has to do with the life sciences and encouraging that industry in our state. The Georgia Global Health Alliance is also a nonprofit that is working to use to push for collaboration among industry, universities, and the private sector, nonprofits, government organizations to work toward global health. And particularly public health in Georgia. At the Institute, our mission is workforce development, and so preparing a future workforce for the life sciences. So that's why we're here, because you guys are doing the same thing, and we want to be able to help you in any way we can. The GHC3 is the Global Health Crisis Coordination Center. And they basically do the things that the CDC can't in times of crisis, because they're able to leverage non-governmental resources. And then the Global Health Innovation District is going to be a physical space that's in the very early stages, and it's down in Atlanta and will be built out over the next few years, but it will house all of this. It should have some really neat space for startups and established companies and lab space and all that kind of stuff. So within the Georgia BioEd Institute, we do workforce development in all those ways that you can kind of see. So we help out with teacher trainings, in particular biotech trainings. We have the Equipment Depot, which some of you guys are familiar with. So anytime you need lab equipment and supplies, before you ever buy anything, come contact us first, because we have stuff for you to keep. Megan's over there waving because she manages it and it's her baby. And she knows all the inventory too. But check with us because there's stuff you can keep. And then we also have a loaning program similar to a library, and it doesn't cost you a dime. We'll ship you stuff or you can come pick it up.
And we'll remind you at the end of this presentation, so.
And then career awareness. So not only for your students and you guys to be able to tell your students what the career opportunities are, but also for those who have graduated high school, either are in college or finishing college or have chosen a different pathway, just to let them know all of the different possibilities that there are within life sciences. We promote BioGenius, and it's a competition. It's an individual competition, research-based. And then finally, Emerging Leaders Network. So if you have any students that have graduated, maybe are in grad school and they're looking to begin a network and getting ready to move into industry, our Emerging Leaders Network supports them and helps them start creating that network of professional support.
Thanks, Kristen.
You're welcome.
All right, so we're going to talk about a couple of the things that we've done as far as trainings go within the past year. So when the pandemic hit, just like everybody else, we had to do a hard swivel and move toward the digital teaching. So the first thing that we did last summer, we got in touch with the Innovate Bio group, which if you're not familiar with them, they are fabulous. They have all kinds of resources, there's an entire site on biotech careers, for example. But they were holding this 5-week summer teacher training. And so we said, we're going to help out with some of the sessions. So we got some real quick kind of toes in the water with teaching virtually for our teachers and doing virtual demonstrations. So first off, Stan Harrison at Morgan County High School presented DNA fingerprinting, and we presented that one in his lab. And then a couple weeks later, he tested positive for COVID, go figure. And so we had to, again, pivot hard. And we hosted the bacterial transformation where he was in his house at home where he was locked in the basement. And I presented from my kitchen, and then we worked with Ying-Su Lo at Baybeck, and she presented from their lab in the Bay Area of California. So we were basically doing 3 different sections of bacterial transformation from 3 different locations. We also talked about the agar art. They have several different plasmids, different colors, so that's pretty awesome. And then Janet Standevan, who's joining us, you'll meet her in a minute if you don't know her already. She presented on her iGEM team and talked about iGEM for this national audience. So we were really happy to be able to get our teachers on this national stage. And the Innovate, basically we met a whole bunch of teachers from across the nation, and it's been a really great networking resource. So I'm happy to connect anybody else who would like to connect with them. So then our training, we were planning this hands-on training called Going Viral. As soon as the pandemic hit, we said, okay, we need to really address the technology that's being used in identifying COVID. And so we had planned it for a hands-on in-person training. That did not happen. So we launched it as a fully virtual training where we sent the supplies home to schools, to their home or their school. So participants received the supplies and they were able to participate alongside our master instructors. So one of our master instructors was Janet Standevan. So she, along with Dr. Brittany Cantrell, they led our training, and we also had some help from Mini PCR. So Allie attended one of those as kind of tech help. Woo. And they actually led a couple of the sessions too when Janet and Brittany were busy with iGEM. So Janet can share a little bit about the curriculum.
Hi, everybody. So the curriculum we covered, we looked at disease detection and did several labs building from using a lab for Mini PCR for going called Central Dogma that we tied into, which literally illuminates DNA to RNA. And so we tied that in with talking about the RNA viruses and the RNA vaccines, which is incredibly timely. And then we went into bacterial DNA disease detection through Mini PCR's food safety there, which was also an introduction to Genes in Space, which is a competition. And I'm sure Allie is going to talk about that. And then lastly, we ended with one I was really excited about, which was qPCR, So we were able to, again, it's another mini PCR lab, and I'm not paid by them. I don't get any kickback, although I wish I did. I get a nice discount on stuff, as we all do, working with Georgia Bio. But the last lab was on qPCR, and so they're able to, by using some interesting reactions, you're able to actually see and quantify the viral load. So you can actually tell when you hit that limit of detection with your PCR. And that was, again, a fantastic lab. And what was, I think, really exciting was then the last day we was an interactive where people were saying they want to do, how did thought this would fit into this unit? What did other people say? And that was a great intermixing. It was fantastic to hear ideas from all over the state and to hear what's going on in the classrooms and also at home this year.
Yeah, and so we're happy to, get you guys into our teacher network so that you can also chat with each other about, what's going on in your classroom. So, and Janet has been great. She doesn't brag on herself enough. She is a former Georgia Biotech Teacher of the Year. Oh, don't hide. And we love her and appreciate her. So thank you, Janet, for joining us this late in the night. Yeah. So with that, I will go ahead and turn it over to Dr. Ali Huang of Mini PCR, and she will walk you through the hands-on component for those who have their materials.
Yeah, thanks so much for the introduction. Just to clear up some logistics, I think a lot of you signed up to receive materials prior to the session. You need to sign up 2 weeks ago so we can get it to you in time. So I think most of you actually have the materials, and we're actually going to do the hands-on Central Dogma Lab. But if you're just tuning in and you didn't sign up ahead of time, no worries. Like Janet mentioned before, this lab is very highly visual, and so you'll still be able to kind of see other people's results and see my results and still get a lot out of it. So if you don't have the supplies, don't worry, don't leave. There's a lot of non-hands-on content that we think is really valuable. Like I said, I'm Dr. Ali Huan. I work for Mini PCR Bio. You've heard that company mentioned a couple of times. We're actually not based in Georgia. We're up in Boston, where we're getting a lot of snow right now. I'm watching it pile up outside my window right now. So we're a small biotech company based out of Boston, and our focus and our mission is to make science accessible for anyone, anywhere. So that we do that through a combination of our low cost, easy to use biotechnology equipment, some of it you'll be using today, as well as really, really innovative curriculum. One of our goals in terms of our curriculum is to bring things to teachers that they haven't seen before. Be able to teach these molecular or biological concepts in a way that you weren't able to before. And one of the examples will be the hands-on Central Dogma Lab that we'll be doing today together. All right, so before I get started, I want to give a really quick overview of the Central Dogma and the processes involved, just to make sure we are all on the same page. I think we have a mix of middle and high school teachers here. The lab is usually used by high school teachers, but we actually had to have quite a few middle school teachers who have used this in their classroom too. So just to make sure everyone's on the same page, I'm just going to quickly run through these background slides. As most of us know, we have DNA. DNA is kind of the blueprint for life. It contains all of the instructions we need to make all the proteins we need in our body, right? So proteins are like what I like to think about are the tools for life, right? You have enzymes that catalyze all these reactions. You have proteins that digest your food. You have proteins that carry oxygen through the blood in your body and provide structural support to your cells. So you have all these different proteins. They all keep us alive and make us who we are, and that's all encoded in instructions found in the DNA. Now, the flow of genetic information doesn't just go DNA directly to protein. There's this middle step in between with RNA. And if you've been listening to the news a lot lately, you might have heard mRNA, messenger RNA, in the news a lot. And the purpose of mRNA is to be the messenger, right? To carry the genetic information from DNA to be able to actually translate that into proteins. And this is a super fundamental foundation of biology. It's so important that it's known as the central dogma of molecular biology, the flow of genetic information from DNA, RNA to protein. Typically, when you're teaching this, especially at the high school level, when you open up your textbooks, students are faced with all these squiggly abstract shapes and diagrams, and it can kind of really get confusing, right? Because it's molecular biology. It's not something you can necessarily see with your own eyes. And so you kind of have to represent them in textbooks with these abstract diagrams. And while there are manipulatives that help students kind of wrap their heads around, okay, this is DNA, this is protein, there is kind of missing this component of real reactions that demonstrate the central dogma. And so that's what we'll be doing today. The key to this is we'll be using fluorescence as our visual markers. For those of you who've never worked with fluorescence before, fluorescence is basically special molecules that when you shine light on them, they emit light themselves. So the example I like thinking of is when you take a highlighter and just scribble on a piece of paper and shine like a UV light or a blue light on it. You've ever been like highlighting bowling, I think, before the pandemic, and then all your stuff glows. That's pretty much it. The UV light or the blue light is hitting the fluorescent molecules in the highlighter liquid and causing it to emit that energy back out as a different colored fluorescent light. And scientists actually use fluorescence in a lot of their biological experiments to track things like where different cell components are in the cell. And so they label all these different proteins with fluorescence, and it makes it super useful for them to see what is going on. And we're going to use these concepts today. We're going to use fluorescence to highlight RNA, to highlight protein, so we can actually see transcription, translation, and the central dogma and process. So just quickly to run through transcription and translation and all that, the analogy that I like using when describing this process is to think of this whole thing like cooking. So your DNA is your cookbook with all your instructions to make the recipes, all the different proteins, all the different dishes you want to make. Each individual recipe is found in a chunk of DNA called the gene. So each gene contains a different recipe for a different protein, all right? And then this whole thing goes through two different processes, transcription and translation. Transcription is the process of DNA to RNA, and translation is the process of RNA to protein. And I'll quickly review these now. But before I do, one thing I want to point out is that transcription and translation usually happens inside living cells. It's happening in all of our living cells right now, but we're all sitting at home or in our classrooms. We probably don't have, incubators and whatnot to be able to grow up cell cultures. And that's where BioBits comes in. So can we actually do transcription and translation reactions without using live cells as our little factory? So what I did is I grew up a bunch of bacteria cells. I busted them open and scooped out all of the components inside, things that we're going to talk about later, like the polymerases and the ribosomes, and made sure there were all the ingredients that we need, right? The nucleotides, amino acids, and energy source, and combined it all in a little tube. And we're going to be using those in a little bit. To make it even easier to work with, we actually freeze-dried these reactions. Freeze-drying is basically when you take something, like I'm using ice cream as an analogy, you freeze it, and then you suck all the liquid out, and it becomes this chalky ice cream that we can send up to the International Space Station for astronauts to eat, and it won't go bad. So we basically done the same thing with these BioBits pellets. We basically freeze dry them into these little pellets that you're going to see in just a bit. And now we can actually just store and ship them at room temperature. We don't have to worry about keeping it too cold. We did have you put it in the freezer just so it stays fine, but it's very easy to ship and store these at room temperature. All right, I'm going to pause there real quick to see if anyone has any questions about the cell-free reactions, because that might be new technology or a new concept for some of you. I just wanted to pause to make sure everyone's kind of on the same page, and we know that what we're using today. And you can always ask later too when we're pipetting.
And we're monitoring the chat as well. So if something comes up later or you feel like you don't want to interrupt or something, feel free to pop a question in the chat.
Yeah. And Yeah, so essentially the freeze-dried cell-free reactions are the BioBits reactions, and basically they're kind of a little factory in a tube that will take whatever DNA that we're going to add to rehydrate the reactions, and it's going to take the DNA, it's going to go through transcription, translation, and make whatever protein that the DNA encodes for. So I'm just going to quickly review transcription, translation. So if we go back to the cooking analogy, let's say you open up your cookbook and you want to copy down a recipe so you can take that down to your kitchen. What you're doing right now, copying that recipe, is transcription. And in this case, the RNA polymerase, which is this green blob in the back, is the molecular machinery that's doing the transcription. It's going to take the DNA, it's going to read it nucleotide by nucleotide and make a copy of it nucleotide by nucleotide, building up this orange molecule called RNA or mRNA, messenger RNA. And that's where the genetic information is being transferred. Now, when we look at the gene, when you're copying your recipe, right, you want to make sure you actually copy the recipe you want. You don't want to accidentally copy another part of a different recipe. So you need to know where to start copying and stop. And usually when you're copying from a cookbook, right, you're just going to copy from the title and go until, you know, the recipe says enjoy or bon appetit or whatever. In DNA, in the gene, there are actually components on the gene that act as start and stop markers for the RNA polymerase. So the RNA polymerase will bind to the promoter, and that's what it knows where to start transcribing. And it'll keep transcribing, copying all this information in the middle over until it hits the terminator, and then it knows how to stop. And then everything in between here, between the promoter and the terminator, will be the genetic information that's found in the mRNA. Okay? So you might be wondering, okay, how are we going to actually see this? Because RNA is not typically visible. And so for this lab, I actually engineered a trick into the gene. So this green component here represents what I'm calling the aptamer sequence. And this is also a trick that scientists use to be able to visualize RNA in their own biology experiments. So how this works is when the aptamer sequence gets transcribed from DNA to RNA, in its RNA format, it will actually fold up on itself, almost like a protein, and form a very specific shape. And this very specific shape acts as a binding pocket and binds to a chemical that I've also freeze-dried into the BioBits. And when this binding interaction happens, it emits a green fluorescent glow. So when we see green, we know that this binding interaction must have happened, and the only way that this can happen is that this binding pocket exists, and the only way that can happen is mRNA exists, which means that transcription was successful. So in this lab, when you see green, you know that means you have mRNA, and then transcription is worked. All right, I'm going to pause real quick to see if anyone has any questions about aptamers. I can go over more detailed questions later, but if you have any questions about the basics of how this works, or what, if you don't understand why green means transcription, let me know now. Okay, and you can always ask later too. I just want to make sure we have enough time for pipetting and everything. All right, so to continue with my cooking analogy, now that you have your recipe copied on your piece of paper, you're going to go down to your kitchen and you're going to read that copied recipe. You're going to interpret the instructions into cooking actions and add the ingredients one at a time. So the same thing in translation. The chef in this case is the ribosome, this light green blob back here. It's basically going to take the messenger RNA, read the nucleotides that make it up, and then translate that into the language of proteins, which are amino acids, using these things called tRNAs, these blue squiggly things. In the interest of time, I'm not going to get into the details here, but if you're teaching this with your students, this is a great opportunity to talk about codons and how the genetic code works and how you translate from nucleotides to amino acids. But for now, all you need to know is that the tRNA, the ribosomes will work together, reading the mRNA, translating that into amino acids, and building up the protein one amino acid at a time, like beads on a string, until the protein is made. And just like the RNA polymerase, the ribosome and the rest of the system also needs its own start and stop indicators. So in this case, they're known as the start codon and the stop codon. So the ribosome is going to start translating at the start codon, and they're going to go until it hits the stop codon. And everything in between here is known as the protein coding sequence, and that's the actual recipe that we're working off. That's where the actual genetic information for building the protein is. And for visualizing proteins, this is a little easier than the aptamer. It's a little more straightforward. There's actually proteins that are found naturally in nature that are fluorescent. The most famous is probably the green fluorescent protein that was discovered in jellyfish in the 50s. But for today, we're actually using the protein coding sequence of a red fluorescent protein that has originally found in coral. And it makes a red fluorescent protein which fluoresces red, as its name indicates. So when we see red, we know that we have our red fluorescent protein, which means we have a protein, which means that translation was successful. So in this case, red indicates successful translation. So again, to recap, green means that transcription worked and you have RNA, and red means that protein worked and you have, or translation worked and you have protein. And you might be wondering, how are we actually going to be able to see this fluorescence? We'll be using this thing called the P51. I have mine right here. If you received materials ahead of time, I'm hoping you were able to build this ahead of time because I sent this out in the email before because there's honestly not enough time for you to build it and do the lab at the same time. So I hope everyone followed my instructions and read the email and built this. But basically what it is, it's a blue light illuminator. If we turn it on, you can see the blue light shine up in the top, and then you stick your tubes in, and anything that's fluorescent in the tubes will light up. It's really cool. As an aside, does anyone know why we call it the P51? I know some of you guys do. So the P51, P stands for photograph. It's photograph 51. It's named for Rosalind Franklin's X-ray crystallography image to help determine DNA structure. As you actually open this, there's a little cartoon depiction of her working away at her microscope. The reason it's called this is because the very first lab we made for the P51 is actually a DNA structure visualization lab. So we decided to name it in honor of her because we feel like she doesn't get enough credit in the scientific community. But since then, the P51 we found has been super useful in a variety of other different labs that all revolve around using fluorescence as a way of visualizing different molecular and biological processes that you normally can't visualize. All right, so I've been blabbing on a lot, but we're going to get to the hands-on portion. So for those of you who do have your supplies, now is the time to get everything together. For those of you who don't have supplies, no worries, please stay on. I'm going to be talking about lots of stuff later, and if you have any questions, you can ask me while everyone else is pipetting. But for those of you who are following along, make sure you have these three things that we sent to you. Make sure you have your P51 that you've assembled and has the battery and it's assembled with the orange filter in place. I'm pretty sure the P51 comes with two filters, an orange one and a yellow one. Make sure you're using the orange one because these results look best under orange. You'll also have your mini pet, your fixed volume micropipette with a bunch of tips. And you'll also should have your reagents. And if we zoom in on the reagents, you should have a vacuum sealed pouch with a strip of little tubes, and in each of these little tubes are the BioBits pellets. You actually only need 4 of these tubes, only need half of them today. So what you can do is you can save your remaining tubes in an airtight bag with this orange desiccant card to keep it fresh. Just stick that back in the freezer, and you can actually use you'll have enough reagents to be able to do this lab again if you wanted to demo it for your students or anything like that, or have another teacher try it. You should also have 4 tubes in another bag. That's just your water, the different DNA, and antibiotic called kanamycin that I'll talk about in a little bit. Oh, the arrows are animating incorrectly. There we go. All right. So if you're missing anything, just let me know. But before we do anything, I'm just going to give you a couple of tips about how to add the reagents to the pellets. When you take your tubes out, before you open the caps, make sure you tap the tubes and make sure the pellets are at the bottom. And you can actually use your P51 as a little tube stand if you don't have a tube stand to help the tubes stay upright and the pellets stay in the tubes. Label the tubes 1 through 4 with a Sharpie. If you don't have a Sharpie that's fine tip enough, my tip is I usually just shove a dot on the first tube, so I know that's tube number 1, and then it's tube 1, 2, 3, 4. And make sure you label the sides of the tubes, not the caps, because if you label the caps, then you take the caps off and you forget which tube to switch. When you do start adding the reagents, don't do it yet, but when you do start adding the reagents, you're going to open the caps one at a time very carefully. You don't want to just rip the caps off, right? Because then you'll lose all the pellets, they'll go flying. So just gently open them one at a time. And when you do add the reagents, don't touch the pipette tip to the pellet. I would actually touch it to the side of the wall right above the pellet and let it drip down. Otherwise, your pellet might get jammed in your tip. And then I wouldn't use the second stop. So for those of you who haven't used a micropipette before, when you press up and down on this button at the top, when you press down initially, you're going to feel resistance, but then you can push past that. The initial resistance is called first stop. And then when you push past that, it's called second stop. Second stop is usually used to make sure you get all the liquid out of your tips into the tubes. But for these small volumes that we're using, that can cause air bubbles. It's not that important to get every little drop out from your pipette tip. So in this case, I would just use first stop. Everyone who's doing the lab, has everyone used a pipette or at least has practiced with food coloring or water beforehand? All right, I can't really see everyone, but I'm seeing some nods and I'm not hearing anyone panic. So I think that's a good sign. All right, so this is what's going to happen. Don't get started yet. I'm just going to run through what's happening in each of these tubes. Tube number 1, you're going to be adding 8 microliters of water. Now your fixed volume pipette is a 4 microliter pipette, so this thing only pipettes 4 microliters. So for tube number 1, you will pipette water twice using the 4 microliters water to get 8. It's also very important every time you pipette and go into the tubes to always use a fresh pipette tip. So when you pipette the 4 microliters of water and you touch the tube, you would have to dispense of your tip, get a new one to pipette the second 4 microliters in, because you don't want to contaminate your supplies, right? To number 2, you're going to add 4 microliters of DNA-A and 4 microliters of water. DNA-A is that contains the gene that I just showed you, right? The gene that has the promoter and the terminator and the aptamer sequence, as well as the start codon, stop codon, and the protein coding sequence. To number 3, you're going to add 4 microliters of DNA-A, as well as 4 microliters of kanamycin, which is, like I mentioned, an antibiotic. And I'll give you a hint now. The way this antibiotic works is that it interferes with ribosomes. So I'll let you think what might happen in that tube. And tube number 4 is actually our mystery tube. I'm not going to tell you what DNA-B is. You should be able to, based on what I told you, make hypotheses or predictions of what you might see in tubes 1, 2, and 3. And we're going to use the results of 1, 2, and 3 to infer what happened in tube 4. It's our inquiry tube. So I'm going to let you go. You're going to add these reagents to the tubes. And once you're done, put the caps back onto the tubes, and we're going to actually heat them for 37 degrees for a few minutes. I say 15 here. That's just an approximate. You don't have to set a timer or anything. The longer you incubate them, the brighter they get. And usually I just use body heat if you don't have a 37-degree incubator. I usually just honestly just stick them in my pocket or kind of stick them in my armpit wherever it's warm. My hands seem to be cold, so that doesn't work very well. And yeah, so I'll let you guys pipette. I'll shut up. And I'll see if anyone, if you guys are just watching, you're not pipetting, let me know if you have any questions. I'm happy to talk about all this stuff. This is all stuff that I actually worked on at MIT when I was doing my PhD there. My PhD was focused on using these freeze-dried cell-free reactions for educational purposes specifically. So I have a lot of experience using these. Happy to talk all day about it. But I'll let you guys go and have fun. I'm going to leave the screen up for you guys, but I'm going to try to, on my other monitor, get more people on the video that I can see in case people need help. Wish this was easier to do on Zoom. I'm assuming Megan and Kristen, you might be able to see more people and can let me know if people are panicking or may need help.
Most
I
people
don't see any panicking.
their
Okay,
videos
good.
off.
All right. And yeah, if you have any questions, feel free to unmute and shout, or you can just put it in the chat if you don't want to be as dramatic. But yeah, I'm happy to just, I guess, sit here for the next 5 to 10 minutes. Again, if you're just observing and you don't have the materials, feel free to ask me anything about this lab, about running this lab remotely We've done it with a lot of different schools. We've had teachers do everything from kind of doing the demo and the students watch to teachers actually sending each of these home with their students, and everyone does it together over Zoom. Kind of depends on your resources and your bandwidth and how much time you're willing to put into it. But we've seen a lot of great ways people can use this lab to teach the central dogma. We actually recently wrote an add-on to our current student guide. So by the way, all of these materials, all the curriculum, the teacher guide, student guide, classroom slides, free webinar demos are all free on our website to download. I will actually pull this up and put it in the chat. You can actually read through the entire lab guide, and we don't believe in selling curriculum. You can actually read the lab guide for free and see how it would fit in your class. But we actually wrote recently a add-on to the student guide where it focuses the lab on mRNA vaccines. So basically using this lab and the visual results of this lab to explain what mRNA is and how that works specifically in the mRNA vaccine. So I know a lot of teachers are looking for resources right now to explain these concepts to their students so they understand how the vaccines work. In the Central Dogma Lab, we've been told by teachers it's a really great way to kind of visually demonstrate how transcription works and how translation works and why that's important for the vaccines.
So Allie, you have a question.
I do, sorry, I have a million windows open and Zoom is not very good at
Can you talk
timing.
more about the aptamer? So how is it engineered?
Yeah, so aptamers are actually something that's naturally found in certain bacteria, usually bacteria, and it's kind of very similar to the fluorescent proteins, where scientists discovered this in nature and realized it was a really valuable tool as a visual marker to be able to track different things happening in their different biology experiments. So I didn't specifically genetic engineer this particular aptamer, but I did take the aptamer sequence and clone it into the gene, so it was right next to the red fluorescent protein gene. So I guess, yeah, I didn't create the aptamer sequence myself, but I did splice it together in the DNA so you could use it specifically for this lab. And again, these naturally evolved in bacteria, and scientists actually have genetically engineered them to make them brighter, to make them more stable. The reason why we're asking you to incubate these reactions at 37 degrees is because the aptamer is most stable at 37 degrees. It would still work at room temperature, but it's brighter at 37 degrees because the aptamer prefers to be in its binding pocket at that temperature. Yeah, they're really, really cool. Scientists have used them in a lot, not just for fluorescent outputs, but in different biosensors and genetic circuits to get them to do really cool input-output sensor things. And there's actually, in the lab, if you read through the lab guide, there's actually a whole section at the end, reading extension for students who are interested in how the aptamer works. And we actually give the sequence of the aptamer so the students can actually see where the nucleotides in the RNA would want to complement to each other and bind and form that binding pocket. So that's a pretty cool exercise to take your students through if they're curious. Are there any non-coding genes included in the DNA samples? So these are, let's see, let me think. So the gene that I showed you is just all the important parts. For the purposes of production, it is a plasmid. So there are some plasmid specific parts just for production's sake, but they're not relevant to the lab. It's got its origin and replication, all that kind of stuff that allows us to, mass produce this. Yeah, no problem. I'm not rushing anyone, but I'm just checking in to make sure everyone is plugging along on pipetting. No one's having too much trouble, hopefully. And so what's going to happen here in a bit is once everyone is close to being done. What we're going to do is for those of you doing the labs, you're going to incubate the tubes. And while we wait for the tubes to incubate, I'm going to go through and actually talk about our Genes in Space competition, which is a free science competition that has now opened and is open through April. And just basically tell you what that is and how you can use it as part of your remote teaching for this year. So that will be coming soon. So I think I'm going to give everyone maybe 5 more minutes to pipette. Is that enough time? And don't worry, you can hopefully pipette and listen to me talk if I move on. If you need way more time than that, feel free to just pop it in the chat and let me know if the majority of you need more time. It's really hard for me to judge how long it takes people to pipette. Where can we get the equipment from? Yeah, so I just linked the BioBits Central Dogma Lab up here. I'll link it again in case you guys missed it. But from here, you can find, so that's the reagents. We sell everything separately just because the P51 is reusable. So you can use it for tons of other labs. So we didn't want to make you buy it over and over again. So let me find the P51 and link that. We also sell pipettes. This lab is usually done with traditional variable volume micropipettes, but we just sent you the fixed volume ones because those are cheaper to give to individual students. There we go. So that's our P51. And again, all of our curriculum is available here, and you can find a bunch of other equipment. I didn't even mention that the reason we're called mini PCR is because our very first product was a miniature PCR thermocycler. That's about this big. Oh, mine's in the other room, but it's about this big, kind of only this big, and it's a lot cheaper and a lot easier to use than a traditional thermocycler. So we actually have a lot of labs around students being able to amplify DNA and then analyze DNA on gel electrophoresis units that we also have that are easy to use. But that's another presentation for another time. I think we have someone demoing. Oh, yep, Janet's showing off her mini PCR. Yeah, so you just run it from your app. We actually have a presentation tomorrow where we'll be demoing some of this equipment. Let me see what it's called. My coworker is going to be demonstrating, it's a session tomorrow at 8:20 p.m. Eastern time called Using Forensic DNA Analysis to Exonerate the Wrongly Convicted. So it's going to be a gel electrophoresis-based lab. My colleague Allison will be talking about that, so tune in. Oh, great to hear that this is fun, Karen. That's what we like to hear. We try to make these a little more fun as not just listening to me blab on for a while. All right, is it okay if I move on in a minute? If you're still pipetting, don't worry, this table can actually be found in the little booklet that you were sent. So when I go off this slide, you can just refer to your booklet to find the numbers that you need for pipetting. All right, I just lost the chat again. There we go. All right, so I'll give you guys 1 more minute, but then I'll just move on and talk. But just know once you're done, cap them back up and incubate them using your body heat, and we'll take a look at them in a bit. All right, so move on to the next slide. And while we're waiting for our tubes to incubate, I'm going to talk about Genes in Space. So Genes in Space is a competition that we run every year. It's for students in grades 7 through 12. And we challenge them to design DNA experiments that look at different things, how biology works in space, essentially. The idea of, people are talking about the idea of space travel, establishing, long-term colonies on the moon or Mars, but we actually don't know a lot about how space travel and microgravity environments impact basic biology. Not a lot has been done on it, surprisingly. And so the purpose of this competition is to ask students to think about some of these kind of basic questions and design experiments that might test about how microgravity impacts all these different biological processes. So here, this is one of my favorite posters that we have, our favorite graphics that we have. At the top, you can see the history of space exploration. Down here, you can see the history of DNA-based research. And here is where you can actually see them intersect with genes in space. And I'll talk about a little bit later, but the very first amplification of DNA, the very first PCR experiment done in space was due to the Genes in Space competition and was designed by a high schooler, which I think is amazing. Yeah, so this is Anna Sophia. She won our very first Genes in Space contest. She proposed studying, I forget what she was saying, but basically she was studying the thing that she wanted to study involved, we were going to have to do PCR in space. And at this point, we didn't know if this was going to be possible. So she proposed an experiment. She won the contest. We sent a mini PCR up into space. This is not a Photoshop picture. It's actually real. There's actually one of our thermocyclers up there, along with her experiment. And we found out that PCR works just as well in space in microgravity as it does back on Earth. And she actually published first author paper as a high school student outlining her experiments and her results, which I think is super, super cool. And we've had a lot of different winners since then. And yeah, so she was testing the effect of cosmic radiation and microgravity on the immune system. We've had so many winners since. Fortunately, I don't have time to go through all of these, but I will point out, for example, the very first CRISPR experiment, gene editing experiment done in space, was done just last year from the 2018 winners. Their experiment was launched in 2019. So all of these high school students are doing super cutting edge research, stuff that has never been done before on the International Space Station. And it all comes from them proposing an idea for this contest. Sorry, my slides are lagging a bit. I can go on. And yes, there's only one winner of Genes in Space every year, but that's just the tip of the iceberg. We have one winner. We have 5 finalists who receive a lot of coaching to get their proposals pitch ready. We have 25 awardees who win equipment for their school, but this comes from a pool of thousands of contestants. And even if your student doesn't win, the experience of applying and thinking about an idea and how to propose that idea and how to design an experiment around the idea is still a very valuable process to go through, even if they don't win in the end. And this is a really useful tool you can use in your virtual classroom. Genes in Space is an ideas only contest. It's free to enter. Students actually don't have to do any experiments to propose their idea. They just have to answer 4 questions on our application form that outline what they want to study, why they want to study it, and it's important, and how they would actually study this in the space. We've had a lot of teachers assign this as a kind of virtual project, and it's worked out really well. So quickly, just to get some logistics, because I know some of you guys are interested in this. The Genes in Space contest cycle, like I said, students write and submit a proposal. They think of an idea, they think about why the idea is important, and they propose an experiment looking at it. From that, we announce all of our awardees. So the contest is now open, and then it closes in April. In May, we announce all our awardees, our 5 finalists. We have our 10 honorable mentions. We have our 5 Junior Scientist Awards. This is open for grades 7 through 12. Typically, the finalists are high school students, but we do want to give middle school students an opportunity to go through the process of applying. So we do award 5 of them the Junior Scientist Award. And we also have 5 Constellation Awards. Constellation Awards, which is for the schools who submit the most applicants. And all these awards get a free set of our equipment, a mini PCR, blue gel system, and a pipette. So our DNA discovery system, a little mini biotech startup for your classroom. The 5 finalists then receive mentoring and coaching. We paired them with Harvard and MIT or Boston-based scientists and researchers, and to help hone and develop their proposal further, so it's ready to be then pitched at the International Space Station Research and Development Conference. They actually pitched these to a panel of astronauts and scientists from industry. It's super cool. And from there, a winner is announced. Last year, we did our first virtual pitch due to the pandemic. And this year, we'll also be doing a virtual-based pitch. The winner then is sent off to space biology camp, where we actually have them do their experiment for real on Earth to get it ready to be shipped to the International Space Station. And then, hopefully in a non-pandemic year, we will fly them out to Cape Canaveral, where they can actually watch their experiment get launched into space, which is a really valuable one in a lifetime experience. So that's a contest cycle. More information can be found at our website. I'll link it in a bit when I'm done talking here. But again, it's for grades 7 through 12. Students can either work individually or in teams up to 4 students. We found for some reason last year, pretty much all the students entered individually because it was just easier to work alone. All the applications need to be sponsored by an adult educator. The online application is now open and will close on April 12th. We have tons of resources. You can go to this website I'm showing here. I'm also going to link it in the chat, genesinspace.org/learn, or check out our YouTube channel. Can I copy these? Nope. Okay, I'll copy them when I'm done talking. Put them in the chat. Our YouTube channel also has tons of videos, including the recorded pitches of all of our finalists from last year, if your students want ideas of what a finalist ready presentation looks like for inspiration. But there's also tons of resources like what is PCR, what is gel electrophoresis, and how do we do this in microgravity. So that is also valuable to look at, and also scoring criteria and all of that. So that is Genes in Space. I know that wasn't 15 minutes, so I'll burn a little more time by pausing now and see if anyone has any questions about this competition. Oh, and the exciting thing I didn't mention is that usually in past years, the competition, one of the requirements was that you actually had to use the mini PCR thermocycler as part of your proposal, as in your experiment needs to involve PCR. This year, we're expanding the toolkit. So this year, your experiment needs to either use the mini PCR to do a PCR amplification, or it needs to use the BioBits freeze-dried cell-free system that you guys just used in your hands-on activity, with the idea of maybe we can express proteins cell-free in space, or it can use more than one, the P51 viewer to visualize fluorescence as a way to highlight different biological processes. So we're expanding our toolkit this year so students can study a wider variety of things instead of just DNA amplification. So I will pause there and see if any of you have any questions about this contest. And while I'm waiting for people to think of questions, I'm going to go find these links and stick them in the chat. I know it's late. People are probably tired. This is close to some of our bedtimes. All right, I'm just going to find I'm going to find the YouTube channel and stick them in here. And then I guess we can move on to our results. But if you have any questions, feel free to ask them at the end as well. Okay, okay, this is the YouTube channel. And again, it's free to enter. You don't need to run any experiments. You just 4 questions in the application. Really great I think it's just great to assign to your students as a project to do, and get them, everyone's excited about space these days. It's a really great way to get them excited about thinking about biology in the context of space. Plus, who doesn't want to shoot their experiment up to the International Space Station in a rocket? All right, not seeing any questions, no worries. Feel free to ask them at any time. But let's review results. It's been about 10 minutes since I cut you guys off. So that should be enough time to start seeing some initial results. If not, I can start going over what we're supposed to see and we can come back to our results and kind of double check. But take your tubes, stick them into your P51 and turn it on. And yeah, if you guys want to turn your cameras on and hold them up to the camera so I actually see your results, that would be really awesome to see. I'm sure you guys are used to telling your students, oh, please turn your cameras on so I can see all your faces. All right, do I see, I don't think I see anyone's yet. Megan, can you see anyone?
No, Kelly's working on it.
Okay. Any of the other teachers who are doing this hands-on want to turn on their cameras and show off their results? If not, don't worry, I have pictures about what they're supposed to look like. So the way this experiment works is after the initial 10 to 15 minutes, you should still start to be able to see the green that indicates successful transcription. Translation happens on the same order, about the same time as transcription, but the red fluorescent protein does take some time to mature. So after seeing the green, you would just leave your tubes at room temperature, come back the next day, and you would have really, really super bright red fluorescent proteins. So I actually, oh, Kelly's holding it up. Can we see it closer to the camera? Can't. It's tilted up, so I can't see anything.
Oh, can you shut the lid, Kelly?
Yeah, you probably want to shut the lid.
Yeah.
Oh, I
see a
see
little bit. See some good glowing.
Yeah. So, yeah. So it looks like you have green in tubes 2, 3, and 4. And I'm just going to show everyone what your results should look like the next day. So the red fluorescent protein is super stable. You can set this up, leave it out overnight at room temperature, or even over the weekend and come back and see the red fluorescent protein. So we can see that there's nothing in tubes 1, 3, or 4, but tube 2 is super, super brightly red fluorescent protein. So let's run over what this means biologically. So tube 1, we didn't see anything, right? Tube 2, we saw it glow green the first day and then red the next day. And I'll go over each of these tubes in more detail to, again, reemphasize what this means biologically. Tube 3, we saw green the first day, but then nothing the next day. And tube 4 behaved just like tube 3, green the first day and nothing the next day. So I'm going to go through tube by tube and quiz you guys on what happened in each of these, okay? So in tube 1, again, we didn't really see anything happen in this tube. In this tube, we just added water, right? There was no DNA in the water that we added. And if we don't have DNA, we don't have transcription, no RNA, no green. And if we don't have that, we don't have translation, we don't have our protein, so we don't have red. Why was it important, though, that we had a tube that just had water in it, even though if the results are boring? Does anyone want to unmute and shout out or put in the chat why it's important to have a water-only condition in this experiment? Control, correct. Specifically a negative control. If you looked at my results from here, if you just looked at tube number 1 by itself, you might think, hey, I think something's glowing in that tube. That's just kind of the way the light reflects off the reaction, reflects off the plastic tube, but it's not anything biological, right? It's just kind of a trick of the light reflecting off. So you use your tube number 1 as a baseline. So you know, okay, this is what nothing looks like. And you can compare all your other tubes to it, so you know if things are actually happening in your other tubes, or if it's also just nothing like the first tube. So negative controls, super important in designing experiments. In all our labs, it's not just about learning the biology behind the reactions, it's also about establishing good scientific method and experimental controls. All right, tube number 2, this is where it gets a lot more fun. So tube number 2, we saw it glow green the first day, and the next day it was red. So let's walk through why. So we know we saw green, so we must have RNA, and we saw red, so we must have red fluorescent protein. For that to happen, we must know that transcription and translation were successful. And for that to happen, we had to add a DNA, which is DNA A, that had the gene that had all of the components that we needed, right? The promoter, the terminator, and the aptamer sequence to be able to visualize the RNA for transcription, as well as the start codon, the stop codon, and the protein coding sequence for the red fluorescent protein, so we can actually see that red fluorescence, right? And like I told you, DNA A contains all of these components, so we were able to see green and then red to indicate successful transcription and translation. Tube 3, we added the same exact DNA that I just talked about, DNA A, but we only saw green the first day and we didn't see red the next day. Again, let's walk through why. We know we added DNA A, which I told you is this gene. So we know that, we saw green and we have RNA, so transcription was successful, but we didn't get red, so we know we don't have protein, and then translation failed somehow. So taking a step back, it's probably one or two reasons, right? It's either something is wrong with the recipe, something is wrong with the gene that I just told you about, or something is wrong with the chef, right? Something is wrong with the molecular machinery involved. It's probably one of the two. Because we added the exact same DNA to the previous tube and that worked fine, we know it's not anything to do with the gene, right? We know the gene is fine, which just leaves the molecular machinery. Does anyone remember what I hinted at the beginning, what kanamycin does? Yeah, stops the ribosome from working. Specifically, it binds to the ribosome and kind of inhibits the mRNA from being able to bind to the ribosome properly, so it mistranslates. So yes, you're correct. In this case, the ribosomes aren't working because of the antibiotic, and you don't get translation. And I think that's super cool because the antibiotic works in very specific ways, right? It specifically inhibits the ribosomes. It doesn't go off and inhibits the RNA polymerase, right? We still got successful transcription. It was specifically targeting translation, and you're actually able to see that visually in your tubes. So that's tube 3. All right, so tube 4, our mystery inquiry tube. Let's figure out what happened here. So in this tube, we saw green the first day, and we didn't see red the next day. Before I dive into this, I'm going to pause here. Does anyone know why the green signal disappears by the next day or is much, much fainter? Anyone have a guess? I'll give you a hint. It has to do with RNA stability. You know, all the news these days about the RNA vaccines and why they have to be kept super cold and why they're super hard to distribute. Why do we have to keep RNA so cold? What happens to it if we don't? mRNA degrades, correct. So basically by the time you come back the next day, the mRNA you generated within the first 15 minutes of your experiment has degraded, so the green signal no longer works. It's honestly actually a great opportunity to tell your students and to discuss RNA stability, great tie-in again to the current mRNA vaccine news stories, that kind of thing. All right, so back to tube 4. So tube 4, it behaved just like tube 3, right? We know we added a DNA. We know we got green, so we must have RNA. So that transcription must have worked. But again, translation failed. So again, going back to our kind of two ways of how translation and not transcription could have failed, something again could be wrong with the gene, or something could be wrong with the machinery. Now in this tube, we didn't add kanamycin or any other reagent, right? And I'm going to promise you here that we didn't spike the DNA B of any like mysterious chemical or anything. So in this case, we know the molecular machinery is fine because we didn't add anything to inhibit it. So it must be something with the gene and how DNA B is a little different. Looking at this gene structure right here, can anyone give me any hypothesis or guesses on what could be wrong or missing from this that would cause transcription to work, but translation to fail? And there's actually multiple correct answers in this case based on the information that you have. So just feel free to shout them out or type them in the chat. Anyone? Okay, yeah, so maybe this protein coding sequence doesn't code for red fluorescent protein. Maybe it's coding for a completely different protein that's not red and fluorescent. That's definitely a possibility. Anything else? So what would happen if there was no start codon? I guess it's too late in the night to make this interactive. If you don't have a start codon, right, the ribosome doesn't know where to start translating. If you took the stop codon and stuck it into the middle of the protein coding sequence, the ribosome will get to the stop codon and stop coding, halfway through. That's called a premature stop codon. The protein coding sequence could be coding for a red fluorescent protein, but maybe there's a mutation in it that causes the protein to fold incorrectly and not work as well. So these are all possibilities. In case you're curious, we just took the whole entire thing out because that was what was easiest to do. But all the different answers you just gave me are correct technically based on the results that you have. All right, so that is the central dogma lab. You saw how DNA was able to go to RNA, to go to protein, the processes of transcription and translation. And by lighting up these processes with fluorescence, right, green for the RNA and red for the protein, you were actually able to visualize this in real time, which is something that, you aren't able to do with other hands-on labs or with paper models or, the abstract diagrams in your textbooks. All right, I think that's the end of my part. I'm going to pass it back over to Megan. But before I do, I wanted to stop and see, does anyone have any questions about this lab, either about the science behind the labs, the logistics of running this lab in your classroom or in your remote classroom? Happy to answer any of those now. I'll also put my email in the chat. So if you have a specific question about how to implement this in your classroom, you can just email me and I can help you out. I've worked with pretty much every possible combination of factors at this point with all the different schools and teachers, so we can figure something out. All right, while I'm waiting for questions, I'll stop sharing and pass this back over to Megan.
Yeah, thank you, Allie.
Thank you. I'm glad you enjoyed this, even though it's almost 9 o'clock.
It's late. Yeah. All right. Well, and on that note, we will not hold you guys too much longer. You can regain a couple nights or minutes of sleep, I guess. So we're just going to talk a little bit more about labs at home. So you guys got to experience doing a lab at home, and Allie provided you with a link so that if you want to do this with your students, you can go online and purchase the materials. So Janet's going to talk a little bit about some other options for how to get your students involved in labs if they are at home.
Hey, so I teach biotechnology, but I have also taught honors bio and environmental science and all of that. And our school has been doing sort of rolling quarantines as we hit the threshold number to send us all home. So the one thing I did in the beginning of the year is I have juniors who are mainly, they're applications of biotech. So they've had a lot of AP Bio and AP Chem. And we did a DNA barcoding unit. And so it was inquiry based, but a lot of them gathered the DNA in their areas, tagged the locations on an app. And then I have, and I'm going to hold them up for you. I made, or I didn't make, but I purchased those DNA Easy sets from Mini PCR. I'm trying to hold this up. And so I had ended up with, I got a grant. And so I ended up with 5 of these sets. And so I could rotate them through the students and they used them, checked them out for 2 or 3 dates. And then they returned them back to me. I had a check off inventory list. And then I restocked the reagents. The great thing about this is the PCR, there are now shelf-stable PCR tabs so that are reaction ready, that you just need to keep them desiccated. And those come from, that are just Taq polymerase. And so I could send PCR reagents home without needing cold chain storage. And then also Mini PCR sells these gel tabs for their gel electrophoresis box, and so, or chamber. And so these gel tabs, literally, they're just small tabs. They make 2 gels, and then the students can either make a 2% gel or a 1.5% gel or a 1% gel, depending on what band length they were going to get. And so this was the highlight. My kids thought they were like the cool in-field scientists because they were getting to extract the DNA at home and then borrow the lab equipment to do it. And it was highly engaging. Now that's like the high-end resource, right? That's the dream world when you have all these resources. But then some other things that I've done is last year when we went on quarantine, so suddenly we ended up doing sourdough experiments at home. And, you know, and this was both setting up and testing experimental methods and experimental design, because then they were using different growth medias for their sourdough and took pictures of it. They all got some really interesting fungi growing in their sourdoughs eventually. But it was a way to keep kids engaged and with just the equipment and the materials that they had at home. Other things that we've used is I use an online subscription called Labster. And they've been really helpful to keep the students sort of at the higher end, working through and thinking through simulations. And that's been really helpful because then when we do get back in the lab, they're ready to go. So I can assign the Labster when we're in quarantine, and then they're ready to jump in for the few days that we're back in lab. So the other thing is for any of the labs that I've ended up having the students do at home, disposal is not a problem. You know, everything can be disposed of in the trash. And the sourdough just needed some household bleach to get rid of some of the mold, but it went really well. So anyway, I'm always happy to answer any questions or work through scenarios. And so I put my information in the chat, and you can please feel free to email me. And speaking about equipment, Megan's going to, I think, talk about all the equipment that you can borrow from the Equipment Depot.
Yeah. So we already spoke a little bit about the Equipment Depot, but again, the depot exists to take supplies from life science companies across the state, and then we get those into the hands of you guys, the teachers, so that you can do hands-on labs. We know that it's expensive, and a lot of this equipment also, you may only use it once a year, so it's a huge investment. So we do have a lot of things like pipettes, gel electrophoresis chambers, incubators that are available for loaning. So if there's something that you're looking for, please contact me. I have people who are like, hey, do you have this kind of shaker? And if I don't have something, I will put you on the list. And so you will receive it when it becomes available. There's a nice picture of Cynthia Greer here at North Hall High School. So I had her down on the list for an incubator, and the second one came available. I went and dropped it off at her school, and I've never seen anybody happier to receive trash. Because literally the company was going to throw it in the trash. So it's a win-win-win. It gets to you guys, and I am happy to do that. I am happy to scavenge companies across the state. And I live really close to the depot. It's in Tucker. It's 8 minutes from my house. So if you do want to come look through the depot, look at glassware, the fridges that we have left, anything, I'm happy to meet you on the weekend as well as during the week. So just feel free to email me if you have anything that you're looking for. So with that, I believe that we are done. So it was great to see everybody on here, and we'll hang out if you still have questions, but otherwise feel free to reach out later. You have our emails, and I'll post those in the chat on Attendify as well, so that they're there after the call.
Or if anyone wants to show off their results, it's been a while, so they're probably even brighter now.
Oh yeah, there you go.
Anyone's brave enough to share their video and show the results, we'll love to see them.
Oh, hi, Elizabeth. We miss you.
Yeah, we'll stick around till 9, I guess. So if anyone has any specific questions about anything that we talked about today, we're happy to talk about it.
And I'll go ahead and stop sharing.
But yeah, if not, thank you so much for joining us tonight. I know it's super late.
Yeah,
But
it
thank
stinks
you.
that we can't do this in person, but
Oh, this is so
because
much
we're-
more fun to do this in person.
but we can have Allie join us from Boston. Who has their pajamas on right now? You're dressed on the top and the bottom, Janet? You're muted, but
I still
nice.
have real pants on.
Oh, nice.
Still have real pants on.
So professional.
I definitely have yoga pants on.
I have the pajama pants on, so.
I'm gonna say goodnight, y'all.
Good night,
All
Janet.
right, thanks for joining us, Janet. I don't know if we just want to stop recording at this point.
Probably. Bye, GSTA.
I see Shawnee and Karen are still on. And I know that Shawnee is our moderator.
Yeah, I'm here. I'm getting ready to head out. Thank you guys so much. This is good information.
Thank you.
Yeah, thank you.
Take care. Have a
You
good
too. Have a good night.
I guess I need to stop recording now.
Oh, didn't I press it?
I don't know. It's still